All right. And so some mitochondrial genes have been identified that appear to be related to the pathology for Parkinson's disease. And so one very active area of research right now is actually looking into some of these genes to try to determine whether or not these, for lack of a better word, sort of maladaptive gene products can be corrected, right? To be able to and I don't really want to go into all of the mitochondrial genes associated with this, but suffice it to say, it's sort of a common thread that we talked about before. So most neurodegenerative diseases, we're starting to find out that there appears to be a mitochondrial component to most, if not all, of these. All right, and so of course, one of the approaches now, all right, is actually in trying to boost the production of proteins, okay, that will act as sort of like mitochondrial checks to repair injury that's happening in mitochondria. So you've got different types of genes that code for proteins that we call cytoprotective proteins. Cytoprotective protein protects cells, right? So cyto means cell and protective. So there are proteins that get released that can actually do things like kill or quench free radicals. So they limit the amount of mitochondrial damage by getting rid of the free radicals and things like that. And so, at least in animal models of PD, some studies have shown that if you actually boost some of these cytoprotective genes, the expression of some of these genes, right? So we boost production of the proteins of genes that code for them, then we can actually prevent some of the pathology associated with Parkinson's disease in animal models of Parkinson's disease. So while this is a far cry from actually proving that this would work in humans, it's a really big and potentially important positive step towards trying to find a better way to improve the quality of life in Parkinsonians, right? So we talked about alpha-synuclein earlier, but just to reiterate, okay, we know that alpha-synuclein is in mitochondrial membranes. It's also present in presynaptic terminals. And so this link between the misfolding of alpha-synuclein and the mitochondrial injury that's happening tend to be focused on by many researchers because these appear to be common threads of neurodegenerative injury in particular, in general, and Parkinson's injury in particular. Right. So how do we treat it? What do we do for Parkinson's disease? All right. Well, because we really have a loss of dopaminergic signaling. Remember, we talked about dopamine neurons in the substantia nigra being selectively eliminated. And again, nobody really knows exactly why that region and that type of neuron are eliminated, but this is just what's been observed in tissues, right? So since we're losing that dopaminergic transmission, we want to do everything that we can to try to boost the level of dopamine signaling, okay? So one way that we can do this is by giving L-DOPA. Now, you might say to yourself, well, why do we have to give L-DOPA? Actually, you might say to yourself, what is L-DOPA, right? So if you go back to the biosynthetic lecture, you'll recall that L-DOPA is one of the precursor molecules to dopamine. So L-DOPA is produced first and then is modified chemically into dopamine, the chemical dopamine, the neurotransmitter. All right. So it's an important precursor to make dopamine. And you might say to yourself, well, why can't we just give dopamine? And there's a couple reasons why you wouldn't want to just give dopamine. First of all, dopamine doesn't really cross the blood-brain barrier. So if we were to administer dopamine the way that we would normally administer a drug to a human patient, right? Because we don't want to drill a hole in the skull, right, and just inject some dopamine in there. That would be a pretty crude and bad way probably of trying to help the patient, right? We might be able to get the dopamine in there, but we'd probably end up causing a hemorrhage or something in the brain. So we want to give something systemically. Maybe we give it orally. Maybe we inject it, okay? But the problem is that it's not going to cross the blood-brain barrier. But what does cross the blood-brain barrier is L-DOPA. And then through normal biosynthetic methods, it can be converted to dopamine and it can actually act at the level of the synapse, all right? So this is one way that we can increase the amount of dopaminergic signaling. Another thing that we can do is we can give agonists. So even though we may not have dopamine around in as large quantities as we normally do in these regions, we can give drugs that bind to dopamine receptors, right? So we can still activate the dopaminergic pathway in the absence of dopamine by making drugs, okay, that are basically chemicals that look very similar to dopamine, have similar three-dimensional structure, and will bind to the dopamine receptors to elicit a response. What's another way we can keep more dopamine around? Maybe we've got some around, we don't want it to get degraded so fast, so let's block the enzyme that degrades it, right? So you might remember that monoamine oxidase is an inhibitor, or is an enzyme that breaks down monoamines. It's going to break down things like dopamine. MAOB in particular is pretty good at breaking down dopamine, all right? So it's just a different form. There's different forms of all of these different enzymes, right? So MAOB inhibitors have been used to try to keep dopamine around a little bit longer in the synapse. And similarly, COMT inhibitors have been used as well, all right? So COMT, this is catecholomethyltransferase, right? So this is what basically converts the L-DOPA into dopamine, or excuse me, L-DOPA has to be converted into dopamine, right? And the COMT will actually prevent the L-DOPA from being degraded before it's converted into dopamine, okay? So if you block catecholomethyltransferase, you're going to have more L-DOPA around for longer, which means you're going to be able to make more dopamine, okay? So this is essentially how these things work. Another technique that's been used is called deep brain stimulation, or DBS. Now, deep brain stimulation literally is electrical stimulation of the brain. This is how it works, okay? So you can put a probe in there, or you can use, there's a few different methods of doing it now, but basically you can try to stimulate these regions to activate the system again, okay? This isn't something that's done often. It's very rarely used. This is actually only for particular cases, okay? And you don't really need to remember these. I just wanted to throw this out there just so that you're aware of what's has been used and what situations it would maybe be used versus not used. So the most common way for sure of treating Parkinson's disease is through these pharmacological agents, okay, these different agonists and inhibitors, right, and L-DOPA, all right. So we're going to round this out by finishing off with neurodegenerative brain injury. So we have neurodegenerative diseases on one hand. So we have diseases that evolve and develop over time, like Parkinson's disease and Alzheimer's disease. They're not typically associated with an injury, right? So it's not like you run down the street, you slip and fall and hit your head, and then 2 days later you have Parkinson's disease, right? Now, to make things complicated, and I'm not going to make it too complicated, but we actually are starting to learn now that having multiple successive injuries to the brain actually could make you more susceptible to developing neurodegenerative disease later in life. All right, this is probably no surprise. It's all over the place now in sports injuries with regards to concussions. Professional boxing has recognized it. The NFL, National Football League, has recognized that repetitive injury to the brain is not a good thing in terms of later outcomes with neurodegenerative disease. So clearly there is a link between these two things. But in terms of brain injury, we're going to talk more about acutely. So what happens right after you get the brain injury versus what's going to happen maybe 20 years later. Okay. So in normal brain injury, so short term, basically you can have a couple different types of cell death. You can have necrotic cell death, necrosis. So this is something I talked about before. This is basically where the tissue breaks down. So you have, let's say you have a stab wound, right? You're going to have a bunch of cells that die. They're going to be broken apart. The membranes are going to break apart. They're going to spill out and they're going to die. Okay. One of the other things that happens, though, particularly in the brain, is when you have a region of cellular injury, okay, you might have a region next to it that's actually healthy. So let's say that your injury occurs right here, okay? All these cells are going to die. This is the focal point. This is right maybe where the initial impact occurred, or the initial injury. But the tissue around it is still viable. But what happens is the injury will spread, okay? Because these cells here actually were forming synapses and connections with neighboring cells. And they were signaling to each other to help to keep each other healthy. And so we call this a detachment-induced cell death. because what happens is these cells that have died are now detached from the neighboring cells, and because these neighboring cells have lost some of that signaling, they tend to weaken. Okay, and then they can die as well as from a loss of what we call trophic signals. So trophic basically just means growth. So they're signals that every cell needs to have normal development and normal growth. Okay, so they can lose some of those. We already talked about apoptosis before, so I'm not going to go over this again, but just know that there are two different forms. Necrotic cell death is normally the immediate form that happens very early on in an injury, and apoptosis is more of a delayed form of cell death. And in fact, some of these cells that have lost trophic signals from those that have died already might actually end up undergoing apoptosis. So some of these cells in the tissues that are a little bit further away from the core part of the injury. All right. And neuroinflammation is basically, we can think of it just like inflammation in the peripheral system, but it's in the brain. Okay. So when dead cells die, they spill out all their contents. All right. And these contents start to get chopped up. All the proteins get chopped up into pieces by enzymes and things like that. And the glia of the brain, the immune cells of the brain, the microglia and the macrophage recognize all these broken pieces of the cells. And they will start to recruit more immune cells to the site, and they'll release signaling molecules called chemokines and cytokines. And those molecules actually recruit more cells to the place to try to sort of like stop the injury from spreading. But what ends up happening is if the injury is severe enough, too much of this inflammatory response can actually cause more injury. And so too much of anything is bad. And so when we talk about neuroinflammation as being a negative thing, it's because overactivation of the inflammatory cascade or system in the brain can actually cause more injury. All right. Astrocytes, they upregulate GFAP, which we talked about before, to kind of wall off an area of tissue around it to prevent the spreading of the injury. Okay, so this is what we call the making of a glial scar. All right. Now, even though the intent is good, they want to wall off the injury, it's bad because it doesn't allow new cells or new growth to occur in the area to kind of try to repair it. So what we can see here, these are micrographs from a study that I performed many years ago. Here you can see this is an astrocyte. This is stained with a fluorescent marker, a fluorescent antibody. And so we can see it's very nice and star-shaped. And here we have, you don't really need to worry about what these words mean here, but this was from a treatment study. So this was just the vehicle alone, the control, and then this was one that was treated with a particular compound. But here we have a core region. It looks black right here because there's actually no tissue. This is a fluorescent marker. So where it's black, there's no tissue here because there was a huge hole. This is where the injury was. And you can see the GFAP positive astrocytes, they get real thick processes and they line the entire outside of this injury, forming a scar, a glial scar, to try to wall this off so that the adjacent tissue around it doesn't start to die as a result of this injury. Okay? Which again is a good thing if you don't want the injury to spread. But on the other hand, it can be a bad thing because now we can't have new cells migrating into the area to rebuild these neuronal connections. All right. Then here we can see another marker. This is an antibody generated against a protein called IBA1, which is made by macrophages and microglia in the brain. So these cells, they look kind of like an amoeba. We call it amoeboid shape because it's round and big. And they become big and round like this when they're activated. Okay. So these are big, round, activated cells. They've been activated by the injury, and they're moving into this site here. So you can see there's a lot of them around the same site. This is the same section of brain. Okay, we just stained this one marker with an antibody that had a red fluorescent tag on it, and this had a green fluorescent tag. So we can look at them both separately. All right. So this is neuroinflammation. These are inflammatory cells responding to the injury to produce chemical signals, to recruit more cells, and to wall off the injury. But in the end, sometimes they actually end up causing more injury as a result. Okay? So what are we going to do? We want to identify the events that cause the injury. We want to try to target selectively which molecular mechanisms cause the injury, and we want to come up with ways to prevent the injury from happening once we know what causes it. All right? So that's it. Thank you very much.